All right, good afternoon, everyone, and welcome to today's Science Luncheon at the Center for Vital Longevity. It is my pleasure to introduce our speaker today, Dr. Bennett, Associate Professor in the Department of Psychology at the University of California, Riverside. Dr. Bennett is a cognitive neuroscience and a former UT Dallas brain health postdoc whose research focuses on understanding learning and memory, how learning and memory develop across the lifespan. She directs the Laboratory of Aging and Neurocognitive Imaging, or LANI Lab, where her team investigates individual and age-related differences in memory by identifying the neural substrates that support learning and retrieval. Her work employs cutting-edge neuroimaging techniques, including high-resolution diffusion imaging and quantitative susceptibility mapping, to explore how brain structure and function change across the lifespan. Today, she'll be presenting some of her work on aging in the LC. Please join me in welcoming Dr. Bennett.
Only she was going to keep it clean, and she did. we go back to our days back in DC.
Yeah.
And I was actually thinking when I was coming out here that it's been 13 years since I left Dallas, but I remember coming to this lunch series when first opened. It's kind of a crazy moment to be the one presenting now. So thanks so much for having me. As my talk title shows, I'm going to be talking about the locus coeruleus today, and it has kind of gotten a lot of attention in recent years. But for those of you who don't know, the LC, or locus coeruleus, is this teeny little brainstem structure. This is approximately where it would be on an MRI scan, and it's about 2 millimeters wide and about 14 millimeters long, and we actually have one in each hemisphere. So it really is a rather small structure. Now, it really came to the interest of a bunch of Alzheimer's disease researchers because it's actually one of the first subcortical nuclei to accumulate tau pathology in the course of Alzheimer's disease. We oftentimes think about entorhinal cortex, and certainly that is one of the first cortical regions to accumulate tau, but LC starts showing some accumulation of tau even before that. Even in the absence of Alzheimer's disease, just in the course of normal aging, we do see some changes in the LC. This graph is from a histological study. It's actually showing cell counts of different types of neurons. I'll direct your attention to the dotted line, which is showing this sort of inverted U-shaped function, maybe more of a plateau of these neuromelanin-containing neurons through about age 75, and then you can see there's a decline after that. But we don't always have to cut your brain open to figure out what's happening in your locus coeruleus. So recent advances in MRI have allowed us to actually image this structure in vivo. The original sequences that were used, the main sequences that have been used, are called magnetization transfer weighted imaging, and they're thought to be sensitive to neuromelanin. So neuromelanin is this pigmented byproduct from the production of norepinephrine as well as dopamine, and that's what this imaging sequence is thought to be sensitive to. And it generates these little hyper-intense spots where that neuromelanin pigment exists. So the arrows are pointing to it just in case you miss it. Again, super tiny. So if we use this magnetization transfer sequence, we can create this contrast, which I'll talk about later. It's a contrast of the signal intensity. And what you can see is, again, we kind of get this inverted U-shaped function, not quite the same as what those early histology studies show, but they suggest that there's an increase in this signal intensity through about age 70. They actually did an analysis to show the inflection is around 57. But again, I would argue there's a bit of a plateau here, and then we start to see a little bit of a decline. So studies comparing younger and older adults have actually shown an increase in this signal intensity metric, thought to be an increase in neuron density up through into old adulthood, and then a decline after that. But there's actually some other approaches that are also known for being sensitive to neuron density among other types of structural changes that might occur in both neurons and glia, and that's diffusion weighted imaging. So this had been largely overlooked by we're interested in structural changes in locus coeruleus, and so it's something that drew our attention. One of the main reasons that we were interested in it is that in looking at some other structures across the brain, we've been able to show that diffusion imaging is sensitive to microstructural changes before you see them in volume. So think of volume as being sensitive to like very late stage loss of neurons, and diffusion was able to capture these much more subtle changes earlier. And we thought the same thing might be happening for diffusion relative to the magnetization transfer. So there's a ton of different ways that we can model the diffusion signal. One of them is that we basically measure the rate of diffusion, which is the jitter of tiny little water molecules. We measure it in a ton of different directions, and then we can summarize that diffusion in an ellipsoid, shown here in yellow. And then we can measure things like fractional anisotropy. How restricted is that diffusion? How cigar-shaped is it? We can also measure mean diffusivity, the average amount, total amount of diffusion in that cell, which is basically how big or small is that ellipsoid regardless of the shape. And then there's some other measures like axial diffusivity, that's diffusion along the primary direction, or radial diffusivity, the average of those secondary axes. And so, whereas fractional anisotropy is really commonly used in white matter, where you have highly aligned, directionally coherent axons, and can make sense of the directional restriction of the diffusion signal, mean diffusivity is actually pretty informative in gray matter and can tell us basically, is there more or less tissue present, right? The tissue is displacing the water. But that's kind of a gross way of looking at diffusion, sort of tissue versus no tissue in this voxel. And so there's been a bunch of different advances, different kinds of biophysical models that have attempted to better model the diffusion signal to tell us something about the underlying physiology. So NADI is one of these approaches. And basically it says, let me take the whole diffusion signal within this voxel, and let me see how much of that total signal I can attribute to this isotropic sphere, and get rid of any like free moving water, like what we would see in ventricles. And then it separates that free signal from we think of as non-tissue dependent from all of the tissue dependent signals, which would be restricted in some way. And then it says, okay, how much of this remaining signal can I attribute to really highly restricted diffusion that would happen within cells, which it models here as a set of sticks. And then basically everything left over modeled as a cylinder is the restricted diffusion that would happen between cellular structures. And so the idea is that these different metrics can tell us something about maybe where exactly the different properties of diffusion are changing. So until about 5 years ago, there hadn't been any studies that had used diffusion imaging to look at effects of age on locus coeruleus microstructure. So that was one of the main questions that we were led to. And then we had some other questions about whether it was going to be stronger for that magnetization transfer rated contrast, whether the age effects were going to be stronger, or the original contrast relative to diffusion, or sky metrics relative to NADI metrics. But it's not just about saying like, hey, no one's done this before, let's apply this new method to the same old thing. What we really want to know is, are these imaging modalities or specific metrics for these imaging modalities more sensitive to effects of age or to effects of pathology? Can we identify which older adults might be at greater risk for LC degradation and eventual Alzheimer's pathology? And the other advantage here is that you need much less specialization to be analyzing diffusion weighted data compared to the magnetization transfer weighted data. It's more difficult. Okay, so in addition to the effects of brain aging on the locus coeruleus, cognitive neuroscientists are interested in its neuromodulatory role in cognition. So the locus coeruleus is the primary source of norepinephrine throughout the brain. You can see the, again, tiny little structure of the brainstem. but it actually projects norepinephrine all throughout cortex and subcortex. And why should we care about norepinephrine? Well, norepinephrine actually modulates arousal, right? So those of you in the back, we're going to start drifting off in about 30 minutes. It's your locus coeruleus that's like, calm down, it's just a talk. We can close our eyes, it's fun, right? It's modulating your arousal and your attention to different things. It actually has, it's a pretty cool structure. It has two different signals. So it has this sort of tonic firing, this background firing whenever you're awake that drops off to nothing sleep. But it can modulate how high or low that tonic rate of firing is. And then it also has this phasic firing. So if something is really need to draw your attention to a stimulus, it can give a phasic burst of firing. And so there's a combination of this tonic and phasic firing patterns that can help direct your attention to. And you don't just have to take my word for it. There's been some really nice demonstrations of this in mice, for example. So they use optogenetic approaches to stimulate cells in the locus coeruleus. And what they're able to do is, check it, they use this fiber photometry approach to actually look at measures of norepinephrine. That's what you see in G and H in the hippocampus. So they're measuring norepinephrine release in the hippocampus in response to stimulation of locus coeruleus. And what you can see is that there's an increase in norepinephrine when you have this 15 hertz phasic activation compared to this very low level 3 hertz tonic activation. And the same thing if you do a 5 hertz tonic, so a higher level of tonic activation, you also get a higher level of norepinephrine. But what's even cooler about this study is that they also looked at the fold response in the hippocampus as well as thalamus, a bunch of other regions. And what they showed is that stimulating LC didn't just dump a whole bunch of norepinephrine into these regions, it actually modulated the fold signal. So you can see that there's an increase in the fold signal for that 5 hertz and increase in that tonic firing compared to the 3 hertz tonic. There's actually a decrease for that phasic burst. And that was seen in hippocampus, thalamus, not in contralateral LC, and not in regions like the amygdala. So it does seem to be a little bit selective.
Do you remember what the x-axis is, for how much time?
I do. Okay, it was a mouse study. They couldn't tell us about the functional consequences of this dump of norepinephrine into these regions. But there has been a bunch of other studies in humans that have shown us that arousing stimuli that we think would be engaging is actually are accompanied by increases in LC activation. So for this one study, this was done just in young adults, and they had them remember scenes. And some of those scenes were preceded by a threat to make them arousing. They were told that they were going to take money away from them, 50 cents, and also a loud bang. So some combination of those threatening stimuli allowed them to seek greater activation to those threatening stimuli compared to non-threatening stimuli in the locus coeruleus. And they were also able to remember those images. So this is consistent with this idea that LC is producing arousal, it produces norepinephrine, and that norepinephrine specifically in the hippocampus, and that can lead to better. There's also some structural evidence that integrity of the locus coeruleus might also be allowing us to remember information better. So this was a study done in both younger and older adults. They also used arousing stimuli, but this was about negative feedback that they got, just negative feedback. And in this study, they showed that the relationship was only significant for older adults. So this is between that magnetization transfer weighted signal intensity on the y-axis and their performance on arousing stimuli. So again, LC does really seem to be able to have an effect on memory, probably via its arousing, its role in arousal. So again, we were saying, hey, what happens with diffusion imaging? Like, are we able to see these relationships? Can very subtle changes in LC microstructure also show this relationship with memory. Is it stronger if we're to choose a memory measure that we know is very hippocampal dependent, or as they had shown in the previous slide, more arousal? So today I'm going to primarily show you data from the published studies, and then I have a little bit of some very work in there, some most recent. The two Langley papers were from the same initial data set that we had. You can see the sample size is a little bit smaller. They differ slightly to had good quality data for the different types of analyses that we ran. And you can see that the outcome measure, the memory measure used was the RAVEL, which I will also describe shortly. The Bennett paper is from a recent grant that we have. You can see the sample size is much larger. We also had neuromelanin sensitive MRI for that one, not just the diffusion sequences. We looked at a word list recall task similar to RAVEL, smaller scale, and then also the mnemonic relation task, which I'll talk about. I only have the MST data in a smaller subset from game 2, which is so in addition to the differences in the sample and the memory measure, I also just want to briefly mention that the diffusion sequence, everyone got a standard 1.7 millimeter diffusion sequence, but the high-res sequence that we used for LC differed between the Langley data set and the Bennett data set. We were able to use a more isotropic, meaning like the same shape, similar shaped edges in our voxel, and sub-millimeter for Langley. We then had a scanner upgrade, and we're no longer allowed to scan at that resolution. And so we had to use a slightly larger voxel for Bennett. The other thing is, because we wanted to model with that multi-compartment naughty model, we needed to have a little bit better SNR. Okay, so let's start with how we tested these first two research questions that are really focused on the effects of age on locus structure. So I'll start with the magnetization transfer weighted data, and this is for comparison to the bulk of work that had been done prior to this study. And so basically what you do when you measure this signal intensity is you measure it within your region of interest, which is shown here in red yellow. This is a standard atlas from multiple magnetization transfer images that we've aligned to the current sample. And then you do it relative to the signal intensity in a reference region, usually within the pons. And then we extracted both an average measure as well as a peak measure, the maximum value, because those are two different values that literature. And what we found is that this signal intensity was higher for older adults compared to larger adults. And we also found that these age effects were magnified for the maximum MTC metric compared to the average metric. And so this fit quite nicely with other studies that used MTC, at least for the ones that have shown significant effects. I might have forgotten to mention that literature is quite mixed, with some studies actually showing no effects of age. We were really interested in doing a broad characterization of age effects, as well as looking at sex differences, whether the age effects vary by subdivision, and by this I mean the rostral versus caudal subdivisions of the locus coeruleus, as well as hemispheric differences. There had been just like one or two papers that reported sex or hemispheric effects, and so we just wanted to give a complete look here. We did find that the age effects were largely driven by males, at least for the average metric compared to the maximum metric. There was also an effect of hemisphere, the right hemisphere showing a stronger age effect. We were a bit surprised to not find an effect of subdivision. There's been some literature to suggest that the rostral subdivision is more vulnerable to cell loss in aging and Alzheimer's, and so we thought we might see a difference here, but just not significant. Okay, so having replicated that pattern of results, we then turned to look at our diffusion metrics. So here again, just aligned that standard mass with our diffusion imaging data. and extracted measures like diffusivity, axial diffusivity, and radial diffusivity. So when we do this, we again found that the diffusivity measures were significantly higher in older adults compared to younger. Now, I will say we were a little intrigued by this because just 2 years prior, we had been reporting the. So in our Langley data set, the initial data set we actually found higher diffusivity in our younger adults compared to our. There are some differences that we thought might explain this. The Langley data set was a little bit older, about 5 years older, and that might have been past that inflection point. They were about 75 years old. Past that inflection point, we do start to see a dip. We accounted for sex in the Bennett paper, which we did not do here in the Langley paper. As I mentioned, we also used those non-isotropic voxels. Originally, I was thinking that it was those non-isotropic voxels that were driving it, but DTI metrics are invariant. They should be invariant to that. So that really shouldn't have been what's explaining the result here. But I think one advantage of our more recent study is that we also had the MTC values in the exact same participant. And so I think this can help us rule out some of the interpretation. So for example, if it is an increase in neuron density, which might be how you would explain the MTC results, that is not what you would, that it could not be explained by our DTI. An increase in neurons is going to increase tissue content of that voxel, decrease the diffusivity, decrease how much water is there. The partial volume effects that from our non-isotropic voxel also wouldn't fit this. That could explain why we see an age-related increase if there's atrophy in older adults and we're getting more ventricle data. That would not explain why we're seeing age-related increase in MTC. So we were able to rule that out. Instead, there's a debate going on in the field about what that MTC measure is actually capturing. Is it just neuromelanin, or could it also be that these neuromelanin-containing neurons have a lot of water, and if there's more of them, they would also have a higher diffusion signal. So I think that that's possible, possibly what. We also found some effects of that the effects of age varied a little bit by sex, a little bit by subdivision here, not so much by hemisphere. I won't necessarily go into those because they weren't very robust, but there were some subtle differences. And then I'll also just mention that we, in addition to looking at those DTI metrics, looked at the naughty metrics and unexpectedly found basically nothing. There were no significant effects of age. They did not really vary by sex. They did not vary by hemispheres. So that more advanced modeling of diffusion in gray matter using naughty didn't really give us an advantage, at least in this individual region. We also did a stepwise logistic regression to figure out which of these metrics is the best predictor of age group. Remember, we're ultimately interested in which imaging modality and which metric is going to give us the best bang for our buck at age effects. And it turns out that it was the maximum FTC metric. So this is the value that gives us about 70% accuracy in saying this person's a young adult, adult. And we were only able to improve that slightly added axial. Yes.
So in all these cases, the definition of LC is guided by this. Can you speak to the individual differences in the localization of LC and whether or not what you're detecting here is differences in
Yeah, so especially for the maximum MTC metric that we pull out, that you really wouldn't think of as being driven by individual differences because you are able, with the neuromelanin sensitive sequence, we can still localize it. So I'm less concerned. And
yeah, so can you repeat what that? I actually got lost on the maximum MTC, maximum metric. What is that actually measuring?
Yeah, So within this region, if we align this to diffusion space, within this region, we find the highest peak signal, which would be in there, we know for sure would be within that individual's locus coeruleus, and then you just subtract it relative to our reference region. So you're basically saying like your LC is somewhere here, but what you're suggesting is that like individuals, their individual LC might be somewhere up and down. And so by using the maximum value, I think that gives us some better assurance that we're getting within that person's, right? If there's a maximum value here, this is where their LC falls, we can pick up a maximum measure here. So we're able to detect it within, I think that measure is a little bit more sensitive to individual differences compared to the average, which you're absolutely right, would be somewhat influenced. But we are also able to look at their individual measures of LC, and like we could have done this at the individual level, and if in PD literature, which is what my colleague is most familiar with, they showed this very similar.
But if you just increase the bounding box of the ROI and just look at the neuromelanin contrast and let the individual subject's image tell you where it is and plot the the spatial distribution of everyone's LCs, does it look like it's falling within the ROI?
Yes.
You know, we've played with the ROI a little bit, and I still can't. To me, it always looks like it's not mapping where it's going to affect it across everyone equivalently.
You certainly have some individuals that it does not do well with, but I would say for the most part, it's one of the more compliant alignments that we've had compared to, even other subcortical structures.
Yeah.
But yeah, that's certainly, so you have been aligning an atlas to NTC intensities and found that it's not what you would define as like.
Not even that far. So when we looked at placement of the ROI in individuals' brain status, it's clear that like there's a lot more variability in just spatial position. For some people, there is a lot of partial volume making.
Yeah, it's certainly something that we can take another look at. It has not strangely been my experience, but that's certainly something that we can take. So you've been like,
you guys have the gold standard scanner, right? You guys can use the neural scanner guide.
Oh, sorry. So that's what you're not doing.
We're not doing that.
Okay, so you're just aligning it to like a functional scan.
We're just looking at all the structure,
For this UPAR.
But you have all the necessary information.
Yeah, I think that's why. But yeah, the other issue is that when you are going, to T1 is one thing, but when you go to any of the EPI sequences like diffusion or functional, your voxel sizes are literally the width of the whole structure at its peak. So it makes me.
You may want to address this later, but since we're looking at neuromelanin, my understanding is that the melanin pigmentation increases with increasing age because it's an accumulative effect. So the reason that you see less pigmentation and smaller signal in younger participants than, say, middle-aged is because it takes time for the melanin to accumulate.
Yeah.
And so there's a huge ambiguity in trying to interpret that inverted U function.
Yeah.
Because you don't know the degree to which you're seeing degeneration of the structure, as opposed to some kind of change in the accumulation of the melanin. So it's not a fantastic measure of integrity of the structure.
Yeah.
I understand it.
And I think that explains perfectly why the histology does not map perfectly onto the magnetization transfer data, right? So the histology shows a plateau through your 60s, your 70s, and only a decline after that.
Yeah.
But the MTC data shows this increase across the younger lifespan, and that would explain after.
Yeah. So people have used magnetization transfer to identify the LC, but a lot of people, I think, would say it's not a good signal of its integrity.
Of its integrity. I see what you're saying.
Yeah.
Yeah.
So it's
all the more reason to use it.
Well, indeed. Yeah.
That's a fair point. Yeah. Okay. Okay, I will just also mention that we, with that original data set, we also wanted to look at not just the locus coeruleus as a structure itself, but its projections. We weren't really comfortable going beyond thalamus. Once the locus coeruleus, the LC neurons project to thalamus, they're going to get lost in there and wouldn't have any confidence that anything coming out of thalamus was those LC projections. So we just limited it to this short tract here, which aligned with the central tegmental tract, and extracted our DTI metrics from this. And just like what we saw in the LC structure itself, we found higher diffusivity in younger adults compared to older. Okay, so what can we begin to say about some of these data? Well, first, yes, it does appear that diffusion-weighted MRI is sensitive to effects of age on locus coeruleus microstructure. We, however, observed both lower and higher diffusivity in younger and younger adults. These mixed findings could be due to some of those differences, methodological differences that I mentioned, or they could reflect sample-specific differences in the underlying neurobiology. I don't have a strong case here to make either way with the data that we have at this point. But I just wanted to show that there's reasons why we might make predictions in both directions. So our initial findings of lower diffusivity could actually reflect higher neuronal density, right? More tissue being present would lower the diffusion signal in adults. One of the interpretations that we are following up on is this idea of neuroinflammation. In early stages of aging, you could have glia proliferating, and an accumulation of glia in the structure could also displace the fluid, or accumulation of other pathologies such as tau, right? So there's a lot of reasons why we might expect there to be a reduction, age-related reduction in diffusion. And there's also a bunch of reasons why we might expect it to be higher in adults. If those neuromelanin containing neurons do have a higher water content, then there being an age-related increase in them would also explain why we have higher diffusion. Same thing for neurodegeneration. If we have a loss of these LC neurons, then we have more room for fluid, and that is going to increase our diffusivity factors. And the other interpretation would be sort of for advanced stage neuroinflammation. Instead of just glia proliferating, now they're activated and swelling, and that would also increase the fluid content, the molecular water content in voxels. So these interpretations could be examined with other imaging modalities, which would be wonderful for us to use to better understand in vivo what's going on exactly. Okay, what about the next part? We did observe that the maximum MTC was the best predictor of age groups, so magnetization transfer did seem to show stronger age effects, although to Dr. Rugg's point, it could very well just have to do with the accumulation of pigmentation over time. Diffusion, the DTI signal for axial diffusivity was the next best predictor. But interestingly, NADI really didn't show any significant main effects. So DTI did a better job, at least within this structure. And again, this can help inform future studies. Maybe don't go so crazy to try and have enough signal for biophysical model when the DTI metrics. Okay, let's shift now. How am I doing? 25 minutes to go. Okay, yeah, it won't. Okay. Let's shift to these last two questions that are really focused on the extent to which the microstructural measures of locus coeruleus are relating to memory performance. So we started with a very general list learning task group. Most of you are familiar with the Raybold. Basically, you present a participant with a list of words, you have them be able to recall them. You do this 5 different times with the same list. You then switch to an interference list B, have them recall it. And then the two key measures we usually look at are the immediate list of recall A following list B, as well as a 20-minute delay. And then there's a recognition choice, forced choice recognition that you do at the end. So as expected, we found that memory was worse in our older adults compared to younger adults. We fully expected for the immediate and delayed recall, which in our hands has been highly correlated with each other. I was a little bit surprised to see that recognition performance also differed between the groups. Usually older adults are good at recognition. And so this might suggest that this language sample that we have some mild impaired adults. Okay, we then examined relationships between performance on this Rayval task and the diffusivity measures. So this is mean diffusivity, radial diffusivity, axial diffusivity. See that they are all showing the same thing, which is, in this case, worse LC microstructure was related to worse. Some of you who do diffusion might be thinking, hold on, lower diffusion is not necessarily worse, but remember, it was the older adults who had lower diffusivity in the sample, so we are interpreting it as worse. The same effects were also seen for both the rostral and caudal subdivisions here. Oh, I should mention that this is only showing the data for our older adults. I meant to put the younger adult graphs in here, but forgot, but there's no significant relationship in. So this relationship between microstructure and performance is only significant for our no effect of rostral versus caudal subdivision. We had predicted that this might have been stronger in rostral and caudal subdivision. And we similarly found that when we looked at the tract that projects from locus coeruleus to thalamus, again, we found significant relationships for only our older adults, not for our younger adults. So again, worse performance is relating to worse diffusion.
Sorry to interrupt again. How do you know that this isn't simply an index of general atrophy or neurodegeneration? Do you have data from other regions which does not show the same relationship? Regions that would be equally sensitive to degeneration?
Yeah, in this study, we also looked at substantia nigra, and I don't believe we saw the relationships that were.
But what if you looked at entorhinal cortex?
In the later study, in the Bennett study, we also looked at volume and showed that volume did not relate to it. So that was just volume of the locus coeruleus.
Right.
And we also have, in some of our, I'll show you that we're starting to look at also like dentate gyrus in our discrimination metric, and we can show that the relationships are actually stronger for the dentate. So I guess that would still speak to this idea that it could be.
Yeah, so the question is, how specific is this? Or is it just that the LC is like a canary in a coal mine? In other words, it's just telling you about degeneration that's relatively diffuse.
Yes, so I think that that's fair to say, but when you say degeneration, do you mean specifically just like cell loss, like atrophy, like global atrophy across the brain?
So I'm talking particularly about diffusion metrics, which are very sensitive to age and quite sensitive to neurodegeneration and impairment. Mm-hmm. So you can see, for example, a correlation in diffusivity within the fornix with memory. With memory performance in older adults.
Yeah. So
my question is, what's special about this? Why should we think this is telling us anything about LC rather than a more general effect?
So I don't recall what other controls we put for these two, the Langley papers. But I will say in all the other data sets that I've worked with, I've controlled for volume in every single one of them, and it does not change the result. So diffusion and volume in my hands are always independent.
Sure.
I think it means regional specificity. So if
I'm
you
talking
drop an ROI elsewhere, brainstem or elsewhere in the brain, You might have a potentially sensitive region here that's actually telling us something about memory impairment really early. And one way of confirming that is to explore relationship to other locations.
Yes, I don't, just trying to keep my Rolodex of evidence here for, I don't believe for this paper, we did substantia nigra for this paper as a control region or a comparison region. I don't believe we had a comparison region, or did, I'm forgetting, did then. And we will be using hippocampus for some of our more recent stuff. So I think that will definitely speak to, we can definitely address that with some of the.
Am I wrong in understanding that part of the question is about staging as well? So you mentioned folks who are being sensitive to certain types of change in age. I learned about the AD model for the brainstem disruption. Is that true in the absence of AD pathology, that it would maybe alter first prior to later regions in cortex, or do they atrophy at the same rate?
I think that's what we need to understand. I think that's the part that's not really known. So there is obviously a bunch of imaging work that's being done now in normal aging in the absence of Alzheimer's disease. And those studies have shown that there is, independent of Alzheimer's disease, age-related absence at LC. But as far as staging, I mean, I would think that it's degenerative just like every other brain. There were some younger, some changes earlier, like 60s compared to the 90s. Okay. In our more recent work, the second data set that we had a much larger battery, and so we ended up using an abbreviated this learning task in which we just had them learn, we exposed them to a single list of 10 items and had them do just immediate recall. And they did this 3 times across 3 different lists. And initially I was really frustrated, like why I chose the 3 different lists. But it ended up being a really great opportunity for us to look at variability in performance, not just mean performance. And the reason that that's informative is because if the locus coeruleus is influencing moment-to-moment arousal, you might expect that it's going to be resulting in variable performance, not just higher or lower mean arousal. So for this study, we did not find any significant age group differences either in the mean amount of items they were able to recall or in variability. Again, remember, this is an easier version than immediate recall. It's just 3 items. There's just 10 items on each list. And I just wanted to plug that this was, I had a lab assistant, Izia Solis, who's now a first-year graduate student at Luncheon, who worked on this data. Okay, we then looked at relationships between list learning performance and these diffusion metrics, and we found that for this, we looked at the whole sample, but independent of age and sex. We found that there was no relationship between the diffusion and mean recall performance. However, there was a relationship between variability of performance and LC microstructure. So lower diffusion, in this case, intracellular diffusion related to worse performance. So again, consistent with this idea that this variability measures might also be for us to be considering. Okay, and then for the last task that we looked at, this is just part of AIM-2. So data collection for this one is ongoing, but we collected data from the mnemonic discrimination or mnemonic similarity task. And essentially what you do is you have participants study a set of objects, and their job is to just say, does it belong indoor or outdoor? You then expose them to different kinds of items at test. So either complete recalls like the piano outlined in green here, to which they should respond old. You show them brand new items outlined in red, to which they should respond new. And then the key trials are these similar lures in which you show them an object that's very similar to, but not quite the same as something they study, which they respond similar. This is an actual diagram of the paradigm that we used. And what we included was this hand grip manipulation during the study phase. So on each trial, they were cued to hold a hand dynamometer at either 5% or 50% of their maximum voluntary contraction. This was a way to induce arousal. So the high hand grip is arousing compared to the low hand grip, which is some prior evidence we've shown with full dilations. And they were tested on the same. So this is work that's being currently led by my graduate student, still a little bit underway. But what we're finding, I should also say this grant is just within. So what we're finding is that just within older adults, older age was significantly related to first discrimination performance, but not to recognition memory. And this represents a large literature by our group others, nothing surprising there. But what was also cool is that we do have some preliminary work showing that mnemonic discrimination was also worse for that low arousal condition compared to the high arousal condition. So the high grip actually in a significantly better discrimination set on trial by trial. Okay, and then again, within these older adults, we examined relationships between LC microstructure and performance, and we did not find any evidence that discrimination related or that diffusion within the locus coeruleus related to mnemonic discrimination, which is what we were really hoping for, but we did find a relationship to dentate gyrus, and there's lots of evidence to suggest that this dentate gyrus region of the hippocampus is specific to this mnemonic discrimination. So hopefully we see something more exciting. Okay, so what can we conclude now? Yes, we consistently found that worse diffusivity in the locus coeruleus related to both worse free recall and variability in our older adults. We're not totally sure why this was only seen within the older adults, not younger adults. We looked at the range of variability within both of our groups, and they're comparable. So it's not like we had some kind of restricted range within our younger adults. But instead, it suggests that maybe that variability is just due to whatever, some neurobiological substrate that's more specific in our older adults. As for the question about whether this relationship is stronger for more hippocampal dependent memory, kind of have to stay tuned. We did find that it was significant for recall, but not recognition in our earlier work, which is aligned with this idea, but we didn't find it for mnemonic discrimination. So we will have to see there. And we did also find that there was some evidence of this as a function of arousal, still evidence. Okay, so what can we take away from this? Well, I can confidently say that the locus coeruleus and its microstructure is an important marker of both brain aging and cognitive aging. I think we have evidence that the locus coeruleus is affected by a number of different neurobiological changes, including cell changes, both increases and decreases, protein accumulation, as well as inflammation. We think that that disrupts the distribution of norepinephrine throughout the brain, including to the hippocampus, and that we have some evidence that this also is driving differences in memory. There's two other points that I just want to make explicit. Most of the time when we think about diffusion-related MRI, we think about it in the context of white matter. But I think we are at a stage now in the field where we can confidently say that it's also really informative about gray matter structures. You just have to make sure that you're using the right metrics, right? FA doesn't necessarily make sense in white matter, but mean diffusivity and other markers really do. And then the last point I want to make is that we really need to be considering the role of these neuromodulatory systems. So locus coeruleus is just one of them. I know a bunch of people in this room are interested in dopamine. There's actually some really interesting evidence to suggest that LC does not just project epinephrine, but so this dopamine, as well as neuromelanin being a byproduct of dopamine. This is not just norepinephrine. So I think that there's interesting questions to be asked. Consider these neuromodulatory systems alone, as well as in the future. And that I will just thank my lab. A lot of this work was done with our MR physicist Jason Langley, as well as of the lab. Thank you guys for your attention.
That hand grip observation is super cool. I've never heard of that. That something that's like widely known. Effective hand grip on memory.
No, there is a well-known link between the effect of hand grip on arousal,
Yeah.
but not necessarily as it relates to. There's some work in the working memory literature, but not long-term.
It's been shown for cold pressure.
For what?
Cold pressure.
Yeah,
Cold
it's the same.
It's the same mechanism.
Yeah, it's the same for like the arousing, the prior studies that it uses emotionally arousing stimuli or threat of taking money or cold pressure. Like all these things are inducing stress or arousal.
Yeah.
And pattern.
Stop sig.
But that
Stop sig.
People have shown that cold pressure is actually associated with increases in corticosteroids and norepinephrine. Not shown directly by assays, blood assays.
Yeah.
So but I'm guessing that you get the same thing with the hand grip.
yeah, we've been looking more at pupil dilation.
I bet it dilates.
Yeah.
So another question about locus coeruleus, which is it's an interesting structure because it accumulates tau throughout the lifespan. It's not only in later life that tau accumulates in LC. It seems to be something that you can see in young adults and right the way through.
Oh, I was not aware of that. It's not just.
And also, there's controversy over whether the LC is an early target of Alzheimer's in terms of tauopathy. Mark Diamond, just across the golf course, he has a paper with Braque actually, where Braque changes one of the Braques, what one didn't die first, where they change their minds. So it's not clear that the LC is an early target of Alzheimer's related tau because the tau is not phosphorylated necessarily. And it's p-tau that's the bad news. And so Diamond and Braque, I think it's in Neuron, have a paper arguing that actually the transentorhinal cortex is the first target for the species of tau that's associated with Alzheimer's. And the tau that accumulates in locus coeruleus may be benign.
Interesting. Okay, I'm involved.
But there's a autopsy literature looking at accumulation of tau in the LC in throughout the lifespan.
Huh.
So the story is I think it's controversial. The story is not.
Yeah. Needs to be yes.
Wondering, kind of two somewhat related thoughts. So first you mentioned that there are different ways to kind of try to different measures or methods to kind of try to get at like more mechanistically what happens in the locus coeruleus with MRI. Like so when they can say a couple words like just get us excited about future direction. Yeah. But also with functional MRI, is it a structure that people typically pinpoint, and what do they find? What are the main ways? Is this way through functional MRI and different tasks to get a better idea of the way by which you have different effects of tonic phasing?
Yeah. Laura Mather's group has been successful with functional activation of LC. Our group has had less success, and that just also comes from less confidence in you're really battling a lot of but they would use the same kind of tasks, so they're just using tasks that use arousal, usually through emotional stimuli, and then showing pupillary correlations to say that arousal stimuli are arousing, they have this result.
Work with Mara was done in collaboration with Emerald Duzel's group at Magdeburg.
Yeah.
I think they're the only group that seem to be able to.
And that would be a
consist-
7 Tesla you need?
I think it was 3T. I think they're the only group that's managed to make that. There's only 150,000 neurons in each
No, that's what I
side.
thought, MRI is 17.
That's the same number of neurons that you have in a typical 2.5 millimeter mitochondria. And they're distributed over half a centimeter. So, and also you've got horrible pulsatile movement artifact. And so you need very, very clever adaptive motion compensation to be able to get a meaningful gold signal.
I thought they were using neuromelanin scans for several years too.
They do. They had a series of those papers. Dorothea Amateur is the person who's led a lot of that research.
Exciting new potential ways to.
Yeah, so one of the, we have not applied this to LC yet, because of the size, but I have recently become really excited about the information. And so we started using diffusion weighted MRS sequences that allow us to look at field specific metabolites and the diffusion properties of those metabolites separate from neuron specific metabolites. And so I think that also, we're trying to target the campus right now, but one day that will help us figure out, like, is there an inflammatory signal? Are we just seeing, are we actually seeing swelling of neurons in LC? Are they are good built, then should be effective signal with them compared to LC. More stuff to come. You'll have to invite me back in 13 more years.
We won't wait that long. Other questions? Do we have any questions online?
We do not have questions online.
All right, thank you.
Thank